coming. It's always an exciting event is to have a defense of any kind, and so an honors thesis defense. This is a combined honors thesis and senior thesis for the School of Earth and Space Exploration with Claire Connett. So Claire has done some really fantastic work over the last year and a half, something like that. Particularly in the last
month.
Longer than that. Since this summer, I think, really working on some enzyme activity, which you can tell us about in detail, and has done a lot of work with Isabel Torres and Alexi Besser. So without more, go ahead and tell us about your work.
Thank you. Hi all, thank you for coming. My name is Claire Connett, and this is my honors SESE project on enzyme-mediated controls on plant litter decomposition across western U.S. drylands. So drylands cover about over 40% of Earth's land surface, and is home to over 2 billion people. And while these regions may seem barren, they're incredibly diverse ecosystems that play a huge role in multiple biochemical processes. So despite their diversity, all drylands share defining characteristics: water scarcity. These regions have low precipitation with high levels of evaporation and transpiration, but drylands are also defined by high spatial heterogeneity, meaning plant communities, soil, microclimate can vary dramatically within a given region. This makes these environments exceptionally vulnerable to climate change and particularly interesting for studying decomposition. So decomposition is a process that drives nutrient cycling through an ecosystem, and as plant litter breaks down, carbon, nitrogen, and phosphorus are released. Some of these are returned back to the soil for plant uptake, and some is lost to the atmosphere. This process is heavily controlled by enzymes, which regulate how efficiently microbes break down organic matter. In drylands, decomposition is shaped by their unique rainfall patterns and patchy plant communities, and this irregular precipitation limits when microbes can actually be active, and while the mix of different plant types creates small areas of distinct litter and soil conditions. These variations influence enzyme activity and how nutrients are able to actually move through drylands. So in decomposition, enzymes are secreted by microbes to facilitate the breakdown of organic matter, cutting molecular bonds of complex compounds to create simpler molecules. These like sugars and amino acids that microbes can then absorb and re-release back into the environment. And enzymes frequently involved in decomposition are hydrolytic and oxidative enzymes. Hydrolytic enzymes break simpler bonds through basically cutting the bonds with water, while oxidative breaks down tougher bonds through reactive oxygen species. And then my project specifically focuses on 5 enzyme types: beta-glucosidase, or BG, and beta-cellobiosidase, or CBH, both which degrade cellulose and contribute to carbon cycling. Beta-N-acetylglucose aminosidase, or NAG, which breaks down chitin and peptidoglycan and contributes to carbon and nitrogen cycling. Phosphatase, or PHOS, which releases phosphate and contributes to phosphorus cycling. And peroxidase, or PROX, which degrades lignin and contributes to carbon cycling. So the problem is most decomposition research comes from temperate woodlands. These regions are often studied more for their lush environments, whereas drylands are seen as desolate wastelands and remain greatly understudied. Temperate woodlands generally have moderate, consistent rainfall, dense tree cover, and moist soils that support continuous microbial activity and relatively fast decomposition rates, whereas drylands have low, unpredictable rainfall pattern, patchy plant communities, and dry soils, making decomposition much slower and highly variable. Because of this, the processes and controls driving decomposition in these regions remain unclear. So my thesis aims to identify what enzymes drive plant litter decomposition in dryland ecosystems. To do this, I asked 3 questions. 1, how does enzyme activity vary across dryland sites? 2, do enzyme activity vary across leaf litter species? And 3, do enzymes show distinct patterns of enzyme-enzyme interactions? And then these questions aim to help me further the understanding of what actually drives decomposition in drylands. So to capture dryland diversity, in 2021, the DEAD project began by selecting 5 sites across the western United States to represent arid and semi-arid regions. Each site has distinct environmental conditions from average annual temperature, total precipitation, and so much more. This provided the opportunity to test how composition varies both within and across dryland types. So while I wasn't involved in the field deployment, understanding the experimental design is crucial. 3 dryland litter species were chosen to represent common dryland litter types. So we got shrub litter or archer, grass litter, boku, and tree litter, purgle. In 2021, approximately 975 litter bags were created and then distributed across all 5 sites. Within the sites, they were distributed between 2 unique microsite conditions, under shrub canopy and in the open areas to capture microenvironment variation. Over the next 2 years, a portion of the litter bags were collected every 4 months to measure and observe decomposition over time. My specific research focuses on the second collection time at 8 months of decomposition. The samples that were collected were then dried, ground, and prepared for further characterization through both pH measurements and enzyme assays. So before measuring enzyme activity, I have to obtain pH. And this is important because enzymes are really influenced by pH. So I got 30 samples from collection 2 and measured them and then modeled them, as you see here. And this figure shows that DBG, Moab, and RCW all have a very similar pH, and then JR and Mojave are kind of like the outliers at 6.25 and 4.63. I then took found the mean of 5.44 so then I could standardize the buffer concentration for actually running the enzyme assays, and then this allows me to compare enzyme potential across the sites while accounting for and seeing pH effects. So after obtaining the mean pH, I was able to calculate absorbance and fluorescence. So assays were done to measure the activity of the 5 previously mentioned enzyme types for 90 samples. Day 1, I prepared a sodium acetate buffer and titrated it to the mean pH of the collection, 5.44, and then next I prepared substrates and standards. On the day of assays, day 2, samples were prepared by mixing them, tearing them with the buffer, and then pipetting them into a well plate along with the buffer, standard, and the substrates. All the well plates were then placed into incubation at 25 degrees Celsius, and activity was measured at 1, 2, and 4 hours after substrates were added. And then from this, I was able to calculate and model the actual activity of the enzymes and then they could be analyzed. So my first major finding of this experiment was that hydrolytic enzyme activity was significantly higher than oxidative across all sites and species. This is an interesting finding, as in temperate regions with moderate and consistent moisture, enzyme activity is a lot more evenly distributed. This suggests that at 8 months of decomposition, drylands are still processing the simpler, easier to degrade compounds. Another interesting finding was that phosphatase activity was the highest. This is the enzyme that helps microbes get phosphorus through the system. And then this suggests that microbes might be prioritizing phosphorus more than carbon or nitrogen. And this suggests 2 things: that phosphorus could be very limited in these regions, and that carbon might be in abundance and less of a priority for the microbes to get. And then while these findings are interesting, further characterization is needed to understand what is really going on. So further analysis showed that plant species was found to be a huge determining factor across all sites. Among the 3 species, purgle showed the highest enzyme activity, while archer and boku were significantly lower. This pattern likely reflects the differences in the chemical composition of each litter type. Purgle tends to have the highest nitrogen content and the lowest carbon to nitrogen ratio, whereas archer and boku have relatively high carbon to nitrogen ratios, making them less nutrient dense and more nutrient poor. Additionally, lignin is another key factor. Grasses like boku often have a higher lignin content, making them a lot more resistant to decomposition reactions, and pergol has a very low lignin content, suggesting that those compounds might make it a lot easier to be degraded in these systems. And then, although litter species clearly shows to be a big influence, other factors may be driving this variation. So further investigations showed that site was found to be a significant impact on enzyme activity. JRN and RCW were found to have the highest overall activity, and DBG having the lowest by far. But the question still remains, what's actually driving these site level differences? So I investigated 3 environmental factors to actually characterize what I think is going on at these environments. So I looked at mean temperature, total rainfall, and litter pH, and that's specifically for this 8-month time period. Among these, litter pH emerged as the strongest driving force of enzyme activity, followed by rainfall, then temperature. CBH showed a positive correlation and a negative to rainfall in the litter Archer, whereas FOS showed the opposite relationship. And then pH showed consistently the most activity in BG, most influence in BG, CBH, and perox, as it was for all species for both BG and CBH. These results suggest that slow decomposing litter may actually be more impacted or more sensitive to environmental variation than fast decomposing litters, and overall pH was the most consistent predictor of enzyme activity, suggesting that even slight changes in litter acidity can substantially influence the decomposition process. Interestingly, the enzyme NAG had no environmental response. This lack of response suggests that additional factors might be at play influencing activities, highlighting the need for further investigation. However, I was able to find that NAG has a relationship to BG, CBH, and perox. This shows that enzymes don't work alone. I found strong correlation between numerous enzyme types, suggesting that microbes are producing these enzymes together to efficiently decompose plant litter. This table shows that BG, CBH, NAG, and perox are all dependent on one another. So then when one increases, you're likely to see an increase in everything else. This shows that this really demonstrates the group effort done by microbes driving decomposition at the microscopic level. So, like most experience, my project does have some limitations. So, while interpreting this data, it's important to consider the limitations of this research. First, mean pH of the site was used, and while this does capture enzyme potential, it doesn't represent the realized or actual activity at the sites. Second, an incubation of 4 hours may be too short to actually measure enzyme activity, as you are likely to see absorbance increase at longer incubation periods. And then finally, not accounting for microsite effect introduces a level of enzyme activity that could not be accurately accounted for in this project. So these findings provide multiple ways to further research. Measuring microsites and their specific environmental conditions like moisture, UV exposure, and temperature would help to further characterize the interconnected forces that drive enzyme activity in drylands. And additionally, expanding the litter types measured would not only help to further the understanding, but then also test these patterns on a broader scale. So overall, my work led to 4 key discoveries. Hydrolytic enzymes significantly outperform oxidative, specifically in the enzyme phosphatase. This tells us that even at 8 months of decomposition, drylands are still degrading easily accessible compounds and could potentially have important implications for carbon abundance in dryland soils. Second, plant litter species is one of the strongest predictors of enzyme activity. This means that knowing the chemistry of what is decomposing is key to understanding how fast it will actually degrade. This suggests that plant species may be a powerful predictor for modeling decomposition rates in drylands. Third, the impact of site was significant. This suggests that decomposition drylands is controlled by a complex interplay of factors, primarily pH, that other decomposition models don't necessarily account for. And fourth, Strong correlations between enzymes demonstrate that microbial communities are producing enzymes in coordinated ways. That this suggests that decomposition does not occur randomly and that microbes may be strategically allocating resources. So, why does this matter? We don't know what's going on with over 40% of our land, and that's why research like this is so important. Even my study at this small scale pointed out that there's something really significant going on here. Our planet is facing unprecedented challenges as climate change threatens to expand drylands through desertification. Understanding how the decomposition process drives nutrients and carbon storage becomes even more crucial. Studies like this are needed to help characterize these ecosystems so we can better predict and protect our planet's future. And then I'd like to thank my committee, and I'd also like to give a special thanks to Isabel for her continued help throughout this project. So thank you all.
Fantastic.
That was awesome, thanks.
So I guess for procedure here, if we have questions from the audience, not the committee, we can take those now, and then we'll ask the audience members to leave, and then we'll grill. The committee will have the chance to grill Claire with all our difficult questions.
I've asked all my questions.
Okay.
It's been so much time.
So many times.
We've had so many, and then she'd be like, wait, I gotta fix this. Okay, that makes it more clear. Let me get that in there. And it's been a lot of conversations. So, a lot.
Nice. So you've become an expert on
Oh, yeah.
enzymes
No,
as well.
this
We
has
should
been
ask you
exciting.
questions.
You'd be surprised. But that's it. You want us to get kicked out now?
Any, Isabel, do you have questions?
I mean, no questions, just really just praise for Claire. Thank you for being so patient and strong throughout this whole process, especially like the testing and trying to set up our parameters for our analyses. It was not easy, and I'm sure you all heard all about it. And I just
Thank you so much.
You're welcome. Okay, yeah, I'm
Okay, great.
I'll turn this off.